Welcome back to the Deep Dive. Today, we're pulling back the curtain on something truly incredible that's quietly revolutionizing everything around us.
Yeah.
Biosensors.
Mm-hmm.
These tiny devices are silently changing the world, from healthcare to environmental protection, and our mission today is to demystify them. We're going to break down their fundamental building blocks, explore how they actually work, and, reveal the incredible ways they translate subtle biological events into clear, measurable signals we can understand.
It's an absolutely fascinating area, because biosensors truly represent this powerful convergence of biology, chemistry, and engineering all working in concert. You can really think of them as miniature detectives. They're meticulously designed to spot specific targets in incredibly complex environments, whether that's a specific sugar in your blood or, say, a pollutant in river water, and then tell us about them in a way that's instantly understandable. Our deep dive today will draw directly from the source material you've provided, covering the core concepts, the underlying principles, and the diverse types of these, ingenious devices. It's perfect for anyone looking to get truly well-informed quickly on this foundational unit of biosensor technology.
So if you've ever found yourself wondering how a tiny strip can tell you your blood sugar, or how a device can detect dangerous bacteria in your food, well, you are definitely in for some aha moments today. We're going to uncover the science behind that everyday magic. Okay, let's unpack this. What exactly is a biosensor, the core concept? All right, let's start with the absolute basics, the big picture. When we talk about a biosensor, what exactly are we referring to? What makes it distinct from, say, just a regular temperature sensor?
That's a great starting point, actually, because it highlights the fundamental difference. At its heart, a biosensor is a device that uniquely combines a biological sensing element, what we call a bioreceptor.
A bioreceptor, right?
Yes, with a physical transducer. Now, the magic happens when that transducer converts a biological event, like a specific molecule binding or maybe a chemical reaction, into a signal we can actually measure.
Okay.
Typically, the signal is electrical, optical, or thermal. And here's the key. The strength of that signal is directly proportional to the concentration of the specific chemical or chemicals it's designed to detect. What's truly fascinating here, the real aha moment perhaps, is that it's not just any sensor. It's a biosensor, precisely because it uses a biological component for recognition.
Okay.
Think about it. Most sensors, they struggle in complex mixtures, right? Like trying to find a needle in a haystack. But a biosensor brings nature's own perfectly shaped magnet, a biological molecule that only sticks to that one specific needle. That's leveraging biology's inherent precision for detection that, no human-made chemical sensor could ever really match in complexity and specificity.
So it really is like a translator, isn't it? Taking this incredibly intricate biological language and turning it into a clear signal we can read and understand.
Yeah.
Maybe a number on a screen or a color change.
Precisely. That translator analogy is perfect.
Yeah.
And the underlying concept, while the applications can seem complex, can actually be broken down into a surprisingly simple plane, a series of essential building blocks that work together to achieve that translation. First, you have the analyte.
The analyte.
Right. This is the specific substance you're interested in detecting, the target molecule. It could be anything from glucose in your blood, heavy metals in wastewater, a particular protein biomarker indicating a disease, or even a virus. The range is, it's truly vast, making them incredibly versatile.
So the analyte is what we're actively looking for. Makes sense. What comes next in this chain after we've identified our target?
Okay, next up is the bioreceptor. This is the biological recognition element, the core of the biosensor. Its job is to selectively interact with that specific analyte. Our sources highlight several crucial types of bioreceptors that we'll dive into in more detail soon, things like enzymes, antibodies, specific DNA strands, or even whole intact cells.
Whole cells, wow.
Yeah, this interaction is the crucial biological handshake, a highly specific binding or a catalyzed reaction that forms the very foundation of the detection process. This is where nature's precision tools truly shine.
And once that biological handshake happens, once the bioreceptor has found its target, how does the device actually read or interpret that event? That seems like the critical bridge, doesn't it?
That's exactly where the transducer steps in. This is the non-biological part of the biosensor, sort of the unsung hero that acts as the essential bridge. Its sole purpose is to convert that subtle biological interaction, that specific binding or reaction, into a measurable physical signal. It takes the biological response and translates it into something like an electrical current, maybe a change in light intensity, a shift in frequency, or even a measurable heat change. Without the transducer, the biological recognition, no matter how specific, wouldn't be comprehensible or actionable for us.
Right. It would just happen, and we wouldn't know.
Exactly.
And then it's all about getting that signal into a format we, the users, can actually understand and act upon, right? Something readable on a device.
Exactly. The final yet equally critical stage involves signal processing and microelectronics. After the transducer generates the raw signal, that signal often needs amplification to make it strong enough to be detected reliably, and then further processing to filter out noise and convert it into an accurate, understandable output. This is essentially the brain of the biosensor.
The brain, okay.
Taking that raw analog data and turning it into something meaningful. A clear number on a screen, perhaps a graphical display, or even an automated alert. So this whole chain analyte, bioreceptor transducer, and signal processing is fundamental to how every single biosensor operates. From a simple glucose strip to a complex environmental monitor, it's a beautifully orchestrated sequence, really. 2, the biological brain. How biosensors recognize.
That makes perfect sense. So we've identified the bioreceptor as the biological brain of the biosensor, the part that does the recognition. Now let's truly dive into those incredible biological elements. How do they bring such, well, mind-boggling specificity to the table? What makes them so precise?
Right. This is where the inherent, almost magical precision of natural biological systems truly shines. It's quite amazing. Our sources outline several major categories of bioreceptors, and each one leverages a unique biological mechanism for incredibly specific and often highly sensitive detection. It's like having a specialized, perfectly fitted key for every unique lock you need to open, you know?
A specific key for every lock. I like that.
Yeah. So first up, we have enzymatic specificity, nature's catalysts. Enzymatic biosensors utilize enzymes,
Okay.
which are these remarkable biological catalysts. In simple terms, enzymes are proteins that speed up specific biochemical reactions. They're like tiny, highly efficient assembly line workers. They recognize and bind to a specific molecule called their substrate, and then facilitate a chemical reaction involving that substrate, which ultimately produces a detectable change.
And they speed things up.
Massively. And the major benefit here is signal amplification. Think of it. One enzyme molecule can catalyze thousands of reactions per second.
Thousands, wow.
Yeah. So a single enzyme molecule can produce a much larger, much more easily measurable signal, even when the analyte concentration is incredibly low. This inherent amplification is why enzymatic biosensors are so powerful. They're often central to some of the most common biosensors we encounter, with the glucose sensor being the most recognized example, right? Helping millions manage their diabetes daily by monitoring blood sugar.
Right, the glucose meter. So the enzyme speeds up a reaction, and that reaction is what the sensor detects, giving us that amplified signal. It's truly ingenious. What about the tools from our own immune system? How are those utilized?
Ah, yes. Good question. Next is the immune system's power, antibodies and antigens in biosensing. Immunosensors are a really powerful subset of biosensors that leverage the incredible specificity of the immune system. They use either antibodies or antigens as their biological recognition elements. The core principle here is the natural ability of antibodies to bind selectively and with extremely high specificity to their target antigens.
Like a lock and key again.
Almost a perfect lock and key fit, yeah. Immunoassays, which are biochemical tests, are fundamentally designed to measure the concentration of an antigen in a sample using this very specific binding reaction. It's truly remarkable how precise this natural recognition system is.
I recall hearing about some of the earliest immunoassay methods being quite complicated, maybe even involving radioactive materials.
Yeah.
How have they evolved to become safer and more practical for biosensors?
You're absolutely right. Initially, methods like radioimmunoassays, or RIA, used radioactive isotopes as markers to detect the binding event. Effective, but they presented safety and disposal challenges, obviously.
Right.
However, enzyme immunoassays, or EIA, were developed as a much safer and highly effective alternative. The key advantage of EIA is that it uses enzymes like peroxidase or alkaline phosphatase as markers instead of radioactive materials. These enzymes catalyze reactions that produce a detectable signal, most commonly a color change, which is not only much safer to handle, but also easily quantifiable.
And easier to see, presumably.
Exactly. What's more, these enzymes also provide significant signal amplification, just like in enzymatic biosensors, because they can catalyze multiple reactions for each binding event. This leads to a much more easily measurable signal, even at very low antigen concentrations. So it's a double win. Safety and sensitivity.
That's a significant leap forward, definitely. And what about detecting our genetic material, DNA or RNA? That seems like an entirely different level of precision needed.
For that, we turn to genetic detectives. DNA and RNA-based biosensors, often referred to as genosensors.
Biosensors.
Yeah. These devices use single strands of DNA or RNA as their bioreceptors. Their recognition mechanism relies on the incredibly precise and specific complementary base pairing or hybridization of nucleic acids. Think of it like two halves of a zipper that only fit together perfectly. A always pairs with T, G always pairs with C.
The base pairs, right?
Exactly. This principle is absolutely crucial for detecting specific DNA sequences, identifying subtle mutations in genes, or even visualizing RNA directly within living cells using clever techniques like molecular beacons.
Molecular beacons,
Right.
what are those?
They're pretty neat. They're designed so that a fluorophore's light emission is quenched, meaning its light is turned off, until it finds and binds to its exact target DNA or RNA sequence, at which point it changes shape, moves away from the quencher, and it lights up, providing a very clear and specific signal.
So it only glows when it finds the right target.
Precisely. And these genosensors are also frequently integrated with nanomaterials, like various nanoparticles, for greatly enhanced detection capabilities, allowing us to find even minute traces of genetic material.
It's astounding how specific biology can be. You even mentioned using entire living organisms or parts of them as sensors earlier. That almost sounds like science fiction. Is that really possible?
It absolutely is. Some of the most intriguing biosensors are built around whole cells and tissues. Living sensors. These are sometimes called biomimetic sensors because they literally incorporate intact living cells, whole tissues, or even isolated cell organelles.
Biomimetic.
Yeah.
Like mimicking biology.
Exactly. They work by mimicking natural biological responses. For example, you could have a taste biosensor that uses actual taste cells which naturally respond to specific tastants, or nerve cells that perceive biochemical signals. Another fascinating example is the use of tissue electrodes for monitoring things like potassium changes directly in myocardial muscle, heart muscle. We also see robust microbial biosensors, which can detect specific targets like methane by utilizing immobilized methane oxidizing bacteria. This approach allows us to tap into the incredibly complex and sophisticated biological machinery of living systems for detection, leveraging their natural sensitivity and response pathways.
So, okay, you've got this incredibly specific biological element, enzyme, antibody, DNA, maybe even a whole cell. But how do you actually attach it to the sensor itself without losing its function or, having it wash away?
Yeah.
That seems like a really delicate and critical step in making a reliable biosensor.
You've hit on one of the most crucial engineering challenges in biosensor development: immobilization techniques. This is absolutely essential for creating a stable, functional, and reusable biosensor. It's not trivial. Our sources describe several key methods for achieving this delicate attachment. The simplest, though often the weakest, is physical adsorption.
Adsorption, just stick it to the surface.
Pretty much. It relies on non-covalent interactions like electrostatic forces or hydrophobic interactions, where the biomolecule simply sticks to the surface. While it's straightforward, it can be reversible, meaning the bioreceptor might detach over time. Not always ideal. For stronger, more permanent attachment, there's covalent binding. This method forms robust chemical bonds directly between the biomolecule and the sensor surface.
Like gluing it down chemically.
Essentially, yes. This is generally preferred because it ensures the biomolecule stays attached reliably, prevents desorption, and often allows for precise control over the number and orientation of the immobilized elements. This control is vital for maintaining the bioreceptor's full functional activity.
Yeah.
You want it facing the right way, so to speak.
Right, so it can do its job properly. And what if you want to keep the biological element encapsulated? Almost like putting it in a tiny cage.
For that, we turn to entrapment. Here, the biomolecule is physically trapped within a protective matrix, typically a gel or a polymer like polyacrylamide or gelatin. Imagine a tiny net or sponge. This matrix is designed with pores that are large enough to allow small molecules, like your analytes, to pass through freely and reach the trapped bioreceptor, but small enough to hold the larger biomolecules securely in place. It's like a permeable protective bubble.
Okay, that makes sense.
Another powerful technique is cross-linking. This uses bifunctional reagents molecules with two reactive ends, like glutaraldehyde is a common one. These reagents chemically link biomolecules to each other, or perhaps to an inert protein like bovine serum albumin, BSA, or directly to a solid support. This creates a robust three-dimensional network or film, really enhancing stability.
Yeah, I've also heard about membranes that come sort of pre-prepared, ready for this process. That sounds like a game changer for speed.
You're thinking of synthetic pre-activated membranes. Yes, these are indeed a significant advancement. They offer a dramatically simplified immobilization procedure, making it much easier and faster to create tailor-made biosensors. For instance, specific polyimide membranes can be provided in a pre-activated form, meaning they already have reactive groups on their surface, ready to go.
So you just add the enzyme.
Pretty much. This allows enzyme coupling to be incredibly rapid, sometimes within minutes, simply by wetting the membrane with a concentrated enzyme solution. After immobilization, bioactive discs can be precisely cut out and then easily mounted onto the transducer or stored for later use. This streamlined approach has really revolutionized the rapid development of biosensors for numerous substrate and enzyme detections. However, despite all these ingenious methods and their many benefits, immobilization isn't without its challenges. It's definitely a delicate balancing act.
What kind of challenge?
Well, sometimes gluing an enzyme down can slightly reduce its affinity for its substrate. It might increase its apparent Michaelis constant, or Km, which basically means it requires a higher concentration of the analyte to work efficiently. It can also decrease its maximum reaction rate, or Vmax, or even alter its optimal pH for activity. These changes are usually due to subtle conformational shifts, changes in the enzyme shape, or changes in its immediate microenvironment once it's fixed.
So it might not work quite as well as it did when it was free.
Potentially, yes. But interestingly, while these might sound like drawbacks, an increase in the apparent Km can sometimes actually enlarge the linear measuring range of the biosensor.
Oh, how so?
Because it takes a higher concentration to saturate the enzyme, the sensor might respond linearly over a wider range of concentrations. So it can actually be an advantage for certain applications needing a wider detection window. It's all about understanding these trade-offs and optimizing the immobilization method for the specific application you need. 3. Translating biology to signals. The transducer story.
That's a fantastic breakdown of how biosensors recognize their targets. Really clear. Now let's move to the next puzzle piece. Once the bioreceptor has recognized its target, how does the biosensor actually turn that recognition into something we can read? Like a number on a display or a change we can observe? This is where the transducer comes in, right? It's the bridge that makes it useful.
Exactly. The transducer is precisely that workhorse, the essential component that converts the subtle biological event, that recognition or reaction, into a measurable physical signal. It's the interpreter, translating the language of biology into the language of electronics, optics, or mechanics. Our sources highlight several major types of transducers, each with its own unique way of achieving this crucial translation. Let's start with the electric current, amperometric, and potentiometric sensors. These are two very common and powerful types.
Amperometric and potentiometric, okay.
Amperometric biosensors are a type of electrochemical sensor that measures the current produced by an electrochemical reaction. The key here is that this current is directly proportional to the concentration of the electrochemically active compound generated by the bioreceptor's interaction with the analyte. A classic example, and a really foundational one in biosensor history, is the Clark-type oxygen electrode.
Clark electrode.
Right. In this setup, a platinum cathode measures the consumption of oxygen during an enzymatic reaction, like the oxidation of glucose by glucose oxidase. As glucose reacts, oxygen is consumed, and the decrease in oxygen current is measured. Modifications to this ingenious design allow for the detection of other compounds like hydrogen, nitrous oxides, carbon dioxide. And many oxidase enzymes, which are widely used, produce hydrogen peroxide, H2O2, as a byproduct. This H2O2 can then be precisely measured amperometrically.
That makes sense. You're essentially measuring the consumption or production of something that creates an electrical current. But I can imagine in complex samples, like blood maybe, there might be other things that also create a current, right? How do they handle that interference?
You're very astute to pick up on that, absolutely. Selectivity can indeed be a challenge. Various organic compounds present in biological fluids like ascorbic acid, vitamin C, or uric acid, they can be co-oxidized at the electrode surface, producing interfering signals.
False positives, essentially.
Exactly. However, this is ingeniously mitigated by using specific semi-permeable membranes, such as those made from cellulose acetate or cellulose nitrate. These membranes act as molecular sieves, basically. They allow only the analyte and reaction products to pass through, effectively blocking larger interfering molecules.
Like a tiny filter.
Precisely. Another strategy is to perform differential measurements. You compare the signal from the sensor with the active enzyme to a control signal from a similar sensor without the enzyme. This allows the background interference to be subtracted out. And current cutting-edge research in this area also focuses on reagentless direct electron transfer systems. The goal here is to shuttle electrons directly between the enzyme and the electrode without needing additional chemical mediators.
Mediators?
Yeah, small molecules that help carry electrons. Getting rid of them simplifies the system and avoids potential issues of mediator toxicity or them leaking into the sample.
Okay, so amperemetric sensors measure a current. What about potentiometric ones? Is it a different kind of electrical signal?
Yes, it's a different aspect of electricity. Potentiometric biosensors, in contrast, measure a change in electrical potential or voltage.
Voltage, okay.
And this voltage change, crucially, is logarithmically proportional to the analyte concentration. This relationship is described by the Nernst law. Common examples include highly sensitive pH electrodes that detect subtle pH changes resulting from enzymatic reactions, like the production of gluconic acid when glucose is broken down. Or you have gas electrodes that detect specific gases like CO2 or ammonia as they diffuse through a gas-permeable membrane. Ion-selective electrodes, or ISE, are also a key part of this category. These are exquisitely sensitive to specific ions in a solution.
Like sodium or potassium?
Exactly. And all solid-state ion-selective electrodes, especially those incorporating advanced conducting polymers or nanomaterials, are becoming increasingly crucial for portable and wearable devices. They offer robustness and miniaturization. For instance, an all-solid-state electrode designed for ammonium ions has been successfully combined with the enzyme ureas for highly accurate urea assays. Shows their practical utility. But, while versatile, these gas-sensing electrodes can sometimes have longer response and baseline recovery times, and they might also respond to other volatile gases present in the sample, which could lead to erroneous readings if not carefully designed. Trade-offs again.
It sounds like there are even more subtle electrical signals that can be picked up. Tell us about conductometric and impedometric sensors. How do they work? They sound a bit more obscure.
Indeed there are, and maybe slightly less common, but very powerful. Moving on, we delve into the hidden signals, conductometric and impedometric sensors. Conductometric biosensors measure changes in the electrical conductivity of a material. Often, this material is a conducting polymer.
Conductivity. How well it conducts electricity.
Exactly. The change in conductivity is caused by the interaction with the analyte or its reaction products. Imagine a polymer whose ability to conduct electricity changes when specific molecules bind to it or when a reaction alters the surrounding chemical environment. The degree of doping, which means intentionally introducing impurities or other elements into the polymer to modify its electrical properties, or even subtle conformational changes in the polymer structure can significantly affect its conductivity. For example, some urea biosensors are based on the precise pH-dependent change in the resistivity, the opposite of conductivity, of a polymer film caused by the enzymatic hydrolysis of urea.
Okay, and impedometric.
Then there are impedometric immunosensors. These measure changes in electrical impedance.
Impedance. That's like resistance, but for alternating current, right?
Right. That's a good way to think about it, yeah. It measures the opposition to alternating current flow. And these sensors measure changes in impedance due to immunological reactions like antibody-antigen binding. This provides a remarkably powerful way to monitor binding dynamics in real time. They are incredibly sensitive to subtle changes right at the electrode surface. As the antibodies bind their targets, they create a nanoscale layer on the electrode, altering the flow of electrons and thus changing the impedance. This allows for label-free detection.
Label-free, meaning no extra tags needed.
Exactly. You don't need to add a fluorescent tag or an enzyme marker, which really simplifies the assay design and use.
This sounds like truly high-tech stuff. Are these the tiny chips we hear about that are revolutionizing electronics, getting down to the transistor level?
Precisely. That brings us to the cutting edge. Field effect transistors, FETs, and carbon nanotubes, CNTs. FETs are certainly at the absolute forefront of miniaturization in biosensing, allowing for incredible sensitivity in incredibly small packages. ISFET-based biosensors, or ion-sensitive field effect transistors, are a prime example. These devices detect minute charge changes at their surface that result from an immunochemical reaction. The ingenious design places antibodies, or bioreceptors, right at the gate region of the transistor.
The gate that controls the current flow in the transistor.
Exactly. When antigen-antibody complexes form, they induce a detectable charge excess right there on the gate, which then modulates the current flow through the transistor, giving us a clear electrical signal. But here's where it gets really interesting and pushes the boundaries of sensitivity. Electrolyte-gated carbon nanotube FETs, e.g. CNT FETs.
Yeah, CNT FETs, carbon nanotubes again.
Yes. These devices utilize semiconducting carbon nanotubes as the channel for electron flow. The beauty is their incredible sensitivity. They're capable of detecting analytes down to picomolar, that's 1/1,000,000th of a mole, and even femtomolar concentrations.
Femtomolar, that's unbelievably low.
It really is. The binding of biological molecules directly influences the charge transport on the CNT surface. The mechanism is quite elegant. When a voltage is applied to the gate, ions in the surrounding electrolyte, like in our body fluids, accumulate at the interfaces, forming an electrical double layer. Now, when an analyte binds to a biorecognition element that's been attached to the CNTs, it changes the local ion concentration and charge distribution. This, in turn, precisely modulates the current flowing through the CNT channel.
So it's like a nanoscale switch where the binding of potentially just a few molecules changes the flow of electricity. That's incredible. How are these tiny things even made?
That's a great question about the engineering aspect. It's quite challenging. These can be fabricated using either random networks of CNTs, which are generally easier to manufacture and offer better device-to-device reproducibility for large-scale production,
Easier.
or aligned networks, which can offer superior performance in terms of signal strength and speed, but are significantly harder to control precisely during fabrication. Getting them all lined up perfectly is tough. What's particularly powerful is that enzymes like glucose oxidase and lactate oxidase can be directly immobilized on the surfaces of these CNTs, forming a highly efficient biosensing interface. This technology is being actively developed for detecting a wide range of critical biomolecules, neurotransmitters in the brain, cancer biomarkers like PSA for early cancer detection, and even for simultaneously detecting multiple ions, moving us closer to multi-panel diagnostics on a single tiny chip.
Amazing. It's amazing how light can also be used in sensing, not just electricity. Tell us about optical biosensors. How do they see what's happening?
Absolutely. Light and color. Optical biosensors are another major class, and they're incredibly versatile. These sensors detect changes in light properties, like absorbance, reflectance, fluorescence, or luminescence, that occur as a direct result of a biological event. Fiber optic biosensors, FOBs for instance, utilize optical fibers to guide light to and from the sensing region. A classic example is a pH optode.
Optode. Like an electrode, but for light.
Kind of, yeah. It changes its optical properties, like color or light intensity, based on pH, much like a miniaturized electronic pH paper using indicators like phenol red embedded in a polymer. Internal enzyme biosensors can also be configured with optical fibers to measure subtle fluorescence changes, like the production of NADH, which is fluorescent. This can then be used to detect compounds like ethanol indirectly. Even more captivating, perhaps, are bioluminescence and chemiluminescence systems.
Like fireflies.
Exactly like fireflies. These are unique because they generate their own light without needing an external excitation source. This often involves specific enzymes like luciferase, which is responsible for the glow of fireflies. This self-generated light makes detection simpler, potentially more sensitive, and highly reliable, as there's no external light source to cause background noise or interference. Bacterial bioluminescent biosensors, for example, are very effectively used for monitoring heavy metals in wastewater. The bacteria's light output changes in the presence of toxins.
Wow, bacteria that glow differently depending on pollution.
Pretty much. And fluorescence-based detection is also widely employed, particularly for DNA and RNA visualization. We briefly touched on molecular beacons earlier.
Right, the ones that light up.
Yeah. To elaborate, these are clever designs where a fluorophor's emission is quenched. Its light is effectively turned off or absorbed by a nearby quencher molecule until it binds precisely to its target DNA or RNA sequence. Once bound, the beacon undergoes a conformational change that separates the fluorophore from the quencher, and then it literally lights up, providing a very clear, very specific signal that can be detected. Another incredibly powerful optical technique is surface plasmon resonance, SPR.
SPR. I've heard of that one.
It's widely used, especially in research labs. This method detects binding events in real time by sensing minute changes in the refractive index near a metal surface, typically a thin film of gold. When biomolecules bind to the gold surface, they change the refractive index right there, which alters how light is reflected off the gold film. This gives an immediate label-free signal.
Label-free again. That seems important.
It simplifies things a lot. And SPR is incredibly sensitive for studying molecular interactions, and its signal can be significantly enhanced by the strategic use of gold nanoparticles. These nanoparticles amplify the refractive index changes, enabling the detection of DNA at incredibly low femtomolar concentrations and greatly improving the sensitivity of protein detection.
And what about sensors that literally feel or weigh things, like nanoscale scales for molecules? That sounds almost impossible.
It's certainly on the cutting edge of physical transduction, but definitely possible. That brings us to the weight and the bend. Mass-sensitive and microcantilever sensors, piezoelectric crystal biosensors, like the well-known quartz crystal microbalance, QCM, operate on a remarkably elegant principle. They use a vibrating quartz crystal. When an electrical field is applied, it vibrates at a very precise resonant frequency.
Like in a watch.
Exactly the same principle. The genius part for sensing is that this resonant frequency changes proportionally to any mass added to its surface. It gets slightly heavier, it vibrates slightly slower. So when an analyte binds to a recognition element immobilized on the crystal, its frequency shifts, directly indicating the mass of what has bound. These AT or BT cut quartz plate specific crystal orientations providing thermal stability are highly stable and incredibly precise for such measurements.
So you're literally weighing molecules sticking to the surface. Amazing.
Effectively, yes. Then we have microcantilevers. These are tiny beam-like structures, often just a few micrometers long, like miniature diving boards. They operate in two distinct modes. In static mode, when an analyte binds to one side of the cantilever, it induces stress. Imagine a tiny force pulling or pushing on one side, causing the cantilever to bend. Downward for compressive stress, upward for tensile stress.
It physically bends.
A tiny amount, yes, detectable with lasers. In dynamic mode, analyte binding changes the cantilever's overall mass or stiffness. This alteration, in turn, changes its resonant frequency, much like plucking a guitar string that changes pitch when you add a tiny weight to it. Both QCM and microcantilevers are exceptional tools for studying protein and nucleic acid interactions and detecting various biomolecules by literally weighing them or sensing the tiny forces as they bind.
And finally, after all these electrical, optical, and mechanical methods, can we even use heat? Can we use temperature changes to detect biological events?
Yes, absolutely, through heat signatures, thermal biosensors. These sensors measure the subtle heat changes, the enthalpy that occur during many enzymatic reactions. Remember, enzymes speed up reactions, and these reactions often either release heat, exothermic, or absorb heat, endothermic.
Right, basic chemistry.
Exactly. The enzyme thermistor is a common configuration. Enzymes are immobilized within a small column, and the very slight heat produced or consumed by their reactions is precisely measured as a temperature change using sensitive thermistors. The advantages of thermal biosensors include their broad applicability, since so many reactions involve heat changes, their suitability for continuous measurement, and crucially, their independence from the optical properties of the sample.
Ah, so good for cloudy or colored samples.
Precisely. That's a huge plus when dealing with opaque, turbid, or highly colored solutions where optical methods might fail completely. However, despite these benefits, they can be quite difficult to miniaturize effectively and integrate into portable devices. They've primarily remained in research settings, with only one enzyme thermistor unit really reaching a semi-commercial stage so far. They require extremely precise temperature control and insulation to pick up such incredibly subtle signals. 4. The science behind the sensor. Principles in action.
It's clear that biosensors are incredibly versatile, using, well, pretty much every imaginable physical principle to detect biological events. But what are the core scientific principles that dictate their performance? What truly makes a good biosensor one that's reliable and effective in practice?
Understanding these underlying fundamentals is absolutely key, yeah. It helps appreciate both their immense power and their inherent limitations, especially when we're evaluating them as robust analytical tools for real-world applications, like in a clinical setting or environmental monitoring. A crucial aspect for many biosensors, particularly those using enzymes, is their enzyme performance, which is often beautifully described by Michaelis-Menten kinetics.
Michaelis-Menten. Okay, that sounds familiar from biochemistry.
Exactly. It's a mathematical model that defines the relationship between the reaction rate of an enzyme and the concentration of its substrate. It helps us understand how efficiently an enzyme works. The Km value, or Michaelis constant, is particularly important here. This constant characterizes the affinity or stickiness between the substrate and the enzyme. Think of it like a lock and key again. A low KMM value indicates a high affinity. That means the enzyme can grab onto and process its substrate very efficiently, even at very low substrate concentrations. This is vital for detecting trace amounts of an analyte.
High affinity, low Km. Got it.
Right. Now, when it comes to the reaction order, at very low substrate concentrations, the reaction rate is directly proportional to the substrate concentration, what we call a first-order reaction. It's like a sparsely populated assembly line. Adding more raw material directly speeds up production. However, at high substrate concentrations, the enzyme essentially becomes saturated. All the enzyme workers are busy. The reaction rate then becomes independent of further substrate concentration. This is a zero-order reaction. The speed is limited only by the enzyme's intrinsic activity, its maximum working capacity, Vmax.
So it's like an assembly line that can only work so fast, regardless of how much material you throw at it once it's maxed out. And we talked about immobilization earlier. Does that affect these enzyme kinetics? Does sticking it down change things?
It absolutely does, and it's a delicate balancing act, as we hinted. While immobilizing an enzyme provides crucial stability for the sensor, it can sometimes decrease the enzyme's affinity for its substrate. This effectively increases its apparent Km.
Increases Km, lower affinity.
Correct, meaning the enzyme might need a higher concentration of the analyte to reach half its maximum reaction rate. This can potentially shift the sensor's linear range. It can also decrease its maximum reaction rate, or Vmax, or alter its optimal pH for activity. These changes are primarily due to subtle conformational changes in the enzyme structure or changes in its immediate microenvironment once it's fixed onto a surface.
So it might not work quite as well as it did when it was free. That sounds like a drawback.
It can be, but not always. Interestingly, this increase in parent KOM can sometimes actually enlarge the linear measuring range of the biosensor.
Oh, how does that work?
Because it takes a higher concentration to saturate the enzyme, the sensor might respond linearly over a wider range of concentrations before hitting that Vmax plateau. So it can actually be an advantage if you need to detect a wider range of analyte concentrations accurately. So it's about understanding these complex interactions and optimizing the system for the specific need.
Okay, so speed is important too. How fast do these devices actually give you a result? And how do we ensure they're only detecting what we want them to and not getting fooled by other stuff?
Those are two critical performance metrics: speed and specificity, response time and selectivity. Response time is simply how quickly the sensor can give a stable, accurate reading after the analyte is introduced. For simpler, single enzyme systems, this can be remarkably fast, often between, say, 45 seconds and 3 minutes.
That's pretty quick. Near real time.
Yeah, fantastic for continuous monitoring. However, multi-enzyme systems, which involve a sequence of reactions, naturally take longer. Each step adds to the overall delay. Many factors influence the speed. The nature and thickness of the membrane the analyte has to diffuse through, the amount of immobilized enzyme, the substrate concentration, even the geometry of the electrode tip, stirring speed, temperature. Lots of variables. Selectivity, on the other hand, refers to the sensor's crucial ability to specifically detect only the target analyte without interference from other compounds present in the sample. This is incredibly important, especially in complex biological fluids like blood or urine, which are, chemical soups.
Gotta find one specific ingredient in a stew.
Exactly that. Antibodies, for instance, are renowned for offering very high specificity due to their natural, highly evolved lock and key binding mechanism. They're designed by nature to be incredibly selective, making immunosensors exceptionally powerful and precise tools for picking out that one target.
So selectivity is key. But how do we get those incredibly low detection limits you mentioned earlier? Down to picomolar or even femtomolar concentrations. That's almost like finding a single grain of sand on a vast beach. How is that possible?
You're right, it's about pushing the boundaries of detection. Often it's all about the power of amplification, improving sensitivity. We need ways to make a tiny signal much bigger. Enzymes themselves are inherently amplifying because they're catalysts. For every binding event, a single enzyme molecule can process hundreds or thousands of substrate molecules per second. This catalytic turnover inherently generates a much stronger signal from what might be a very weak initial interaction. One tiny event triggers a cascade.
The enzyme does the heavy lifting.
It does. Nanomaterials also play a huge role here, as we'll discuss more. For example, gold nanoparticles can dramatically enhance signals in techniques like SPR, leading to orders of magnitude improvement in detection limits, allowing us to sense analytes that would otherwise be completely invisible. And enzymes with a high turnover number, those that process many substrate molecules per unit time, are particularly valuable for providing truly excellent sensitivity.
Makes sense. And finally, how do we make sure these sensors are reliable for repeated use in real-world conditions? Not just a one-time lab experiment, but something dependable.
That leads us to keeping it reliable. Stability, reusability, and interference management. These are critical for practical use. Stability is a key performance parameter. The choice of immobilization methods, which we discussed earlier, is absolutely critical here. The aim is to ensure that the enzyme or bioreceptor remains active and functional for an extended period, sometimes for weeks, months, or even thousands of individual measurements without significant loss of activity. This operational stability is vital.
So it doesn't just die after a few uses.
Exactly. Closely related is reusability. Many biosensors are cleverly designed to be regenerated and used multiple times. This significantly reduces costs and waste. For instance, in piezoelectric immunosensors, after a measurement, the analyte bioreceptor complex can often be dissociated unbound by simply washing the sensor with a mild acidic solution. This effectively resets the sensor, making it ready for a fresh measurement. This reusability is crucial for making biosensors economically viable and environmentally friendly in many applications, moving beyond single-use disposables.
Resetting the sensor. Clever.
Yeah. Lastly, interference management is a constant ongoing challenge, especially when dealing with complex biological samples like blood. It contains countless molecules that could potentially interact with the sensor. Other compounds in the sample can sometimes produce false signals or decrease the sensor's activity, leading to inaccurate readings. To combat this, several ingenious strategies are employed. Using highly selective membranes that act as gatekeepers, only allowing the target analyte through while blocking interfering molecules. Employing differential measurements, comparing the signal to a control without the enzyme to subtract background noise. Or designing systems that can actively exchange internal solutions to clear interferences between measurements. For example, volatile gases can interfere with gas-sensing electrodes, or large macromolecules like proteins in blood can foul electrochemical electrodes by physically sticking to the surface, reducing their reliability over time. Robust biosensor designs actively incorporate these sophisticated strategies to ensure accurate and reliable measurements in real-world scenarios. That's crucial for students learning this unit to grasp the practical challenges. V, the nanorevolution. Tiny tech, huge impact.
That's a comprehensive look at the fundamental principles. Really helpful. This brings us squarely to nanotechnology. We hear about nano everything these days, but how are these incredibly tiny materials specifically changing the game for biosensors? Is it just making things smaller, or is there more to it than that?
That's an excellent question because it's far more than just miniaturization, although that's part of it. Nanotechnology has truly revolutionized biosensing, unlocking capabilities that were previously, pretty much unimaginable. This is the essence of why nano, unlocking new potential. We're talking about nanomaterials, which are typically sized between 1 and 100 nanometers. For context, a human hair is about 80,000 nanometers thick, so incredibly small.
Tiny.
And materials at this scale exhibit unique properties that are vastly different from their bulk counterparts. Things like carbon nanotubes, graphene, quantum dots, various nanoparticles. They offer incredible advantages. First, they provide an increased surface area, massively increased. Imagine trying to immobilize bioreceptors on a flat plate versus a highly wrinkled, porous surface made of nanoparticles. Nanomaterials offer an enormous surface-to-volume ratio.
More places to stick the detectors.
Exactly, providing significantly more space for bioreceptor immobilization, which means you can pack more detectors into a tiny area, leading to stronger signals and higher sensitivity. Second, they have enhanced electrical and optical properties. These materials often conduct electricity or interact with light in ways that bulk materials just don't. This leads directly to higher sensitivity and remarkably lower detection limits, sometimes even down to femtomolar concentrations for DNA. That's just a handful of molecules.
Incredible.
They often offer improved stability, making biosensors more robust, longer-lasting, and challenging environments. And finally, yes, they enable miniaturization, allowing for smaller, more portable, and even implantable devices. This is helping address crucial issues like unreliability, lack of automation, and poor signal transduction that often plagued earlier biosensor designs. Nanotechnology is truly overcoming many of the traditional limitations.
I've definitely heard about carbon nanotubes being hailed as a wonder material. How exactly do these tiny powerhouses fit into the world of biosensors?
Carbon nanotubes, or CNTs, are indeed tiny powerhouses, yeah. And they're right at the forefront of this nano revolution in biosensing. They are essentially hollow cylinders of graphitic carbon, like rolled up sheets of graphene. They possess exceptional structural, electrical, and mechanical properties. They can be single-walled SWCNTs, just one layer thick, or multi-walled MWCNTs, consisting of several concentric tubes. Their one-dimensional nanosize, coupled with their enormous surface area, exceptionally high carrier mobility, meaning electrons can move through them very quickly and efficiently, and remarkable biocompatibility make them ideal for biosensing applications.
What roles do they play?
In biosensors, CNTs are used in several powerful ways. They function as highly efficient nanoelectrodes, enabling extremely localized and sensitive electrochemical detection. They also serve as fantastic scaffolds for enzyme immobilization, creating a high-density platform for bioreceptors. Crucially, they facilitate efficient direct electron transfer between the enzyme and the electrode, bypassing the need for those mediators we talked about, which simplifies the system and improves performance. And most notably, as we touched on earlier, they are integral to electrolyte-gated carbon nanotube FETs, e.g. CNT FETs, for ultra-sensitive detection. Their unique surface charge transport properties are incredibly sensitive to even minute changes in the surrounding environment, making them extraordinarily responsive detectors. When fabricating these, random networks of CNTs are generally more popular due to easier manufacturability and better device-to-device reproducibility, although aligned networks can offer superior performance despite being harder to precisely control in their orientation.
And what about other types of nanoparticles and those intriguing quantum dots? How do they illuminate detection, almost literally?
That's a perfect way to describe it. Nanoparticles and quantum dots. Illuminating detection play a critical role, especially in optical biosensors. Gold nanoparticles, AUNPs, are used extensively, not just because they're biocompatible, but because of their unique optical and electrical properties. They significantly enhance signals in SPR biosensors by amplifying refractive index changes, and they dramatically improve electron transfer in electrochemical biosensors. They are so effective that they can help detect DNA at femtomolar concentrations, pushing detection limits way down, and greatly enhance protein detection, offering remarkable signal amplification through their high surface area and sometimes catalytic properties.
So gold isn't just shiny.
Definitely not at the nanoscale. Then there are silica nanoparticles. These are fascinating because they can be precisely engineered and doped with various fluorescent dyes. This allows them to create highly luminescent labels for the selective tagging of a wide range of biomedically important targets: cancer cells, specific bacteria, individual biomolecules. Compared to traditional organic fluorophores, the standard fluorescent dyes silica nanoparticles offer significant advantages: better multiplexing capabilities, detecting multiple targets at once, easier functionalization, attaching things to their surface, greater sensitivity due to brighter and more stable emission, and superior photostability.
Photostability, they don't fade as fast.
Exactly. They don't bleach as quickly when exposed to light. They also hold immense promise as advanced drug delivery systems, showing their versatility beyond just sensing. Finally, we have quantum dots, QDs. These are truly at the cutting edge. They are semiconductor nanocrystals, typically just a few nanometers in size, that exhibit unique optical properties. When excited by light, they emit bright, pure colors, and their emission wavelength can be precisely tuned by simply changing their size.
Changing size changes the color.
Precisely. A smaller quantum dot emits blue light, a larger one emits red light, and you can tune it across the spectrum. This size-tunable emission is a game changer. They possess significantly higher photostability than traditional organic fluorophores and are utilized in highly sensitive fluorescent biosensors for detecting DNA, microRNA, which plays key roles in gene regulation and disease, various proteins like PSA for cancer screening, and even for visualizing cellular processes like apoptosis or programmed cell death. A key advantage of QDs, beyond brightness and stability, is their distinct narrow emission wavelength. This allows for multiplex analysis. You can use one light source to excite multiple types of QDs, each emitting a different specific color, and simultaneously detect multiple targets within the same sample. A huge leap for diagnostics. Bringing it all together, where biosensors make a difference.
So we've explored the fundamental building blocks, the different physical principles they use, the recognition, the transduction, and the exciting, game-changing role of nanotechnology. But what does this all mean for us? Where do we see biosensors making a tangible, real-world impact today, and what can we expect in the very near future? What's the practical application of all this incredible knowledge?
This is really where the deep dive pays off, right? Connecting the science to everyday life. Biosensors are already making huge strides across various sectors, profoundly impacting our daily lives and driving scientific progress. They're not just lab curiosities anymore. They're becoming indispensable tools. In personal health and medical breakthroughs, biosensors are, well, indispensable. The glucose biosensor is arguably the most recognized and impactful example, enabling millions globally to monitor their blood glucose daily for diabetes management. It's transformed how diabetes is managed, from a cumbersome lab test to an easy at-home routine. The ongoing goal, which is incredibly ambitious and exciting, is to develop continuous glucose and insulin monitoring systems. These could be foundational for truly reliable artificial pancreas systems, potentially freeing individuals from constant manual monitoring and injections.
The artificial pancreas, that would be life-changing.
Absolutely. Beyond glucose, biosensors are crucial in clinical diagnostics. They detect a wide array of disease biomarkers, things like cancer antigens for early detection, viral particles, think rapid COVID tests, various metabolites like lactate, urea, creatinine that indicate organ function.
Kidney function, liver function.
Exactly. As well as neurotransmitters and hormones that impact mental and physiological health. This precise, rapid information aids significantly in disease prognosis, accurate diagnosis, guiding personalized drug delivery, and monitoring treatment effectiveness. Furthermore, the development of miniature biosensors for implantable devices is truly revolutionary. These are designed for in vivo monitoring inside the body of crucial physiological parameters like pH, PCO2, carbon dioxide levels, and potassium ions directly in tissues or blood.
Inside the body. Wow.
Yeah. Imagine real-time brain chemistry monitoring via neurotransmitter sensors or even gut health insights, all delivered from within the body continuously.
So they're not just for personal health, but also for the wider environment. That seems like a massive scale jump from an individual's bloodstream to, say, a river.
Absolutely. Biosensors serve as crucial guardians of our environment. They are vital for pollution monitoring, detecting heavy metals, phenolic compounds, pesticides, and other toxic agents in wastewater, soil, and air. For example, those bacterial bioluminescent biosensors we mentioned, where the light output of bacteria changes in the presence of specific toxins, they can detect certain heavy metal ions with a remarkable sensitivity and speed.
Like a living alarm system.
Kind of, yeah. They're also used extensively for water quality monitoring. For instance, by detecting changes in microorganism activity as a rapid indicator of overall water toxicity, far faster than traditional lab tests, which can take days. Beyond environmental protection, biosensors are integral to ensuring what we eat. They are widely used in the food industry.
That's so.
For monitoring fermentation processes, checking precise concentrations of glucose and alcohol in brewing or winemaking, for example. Ensuring overall food safety by detecting pathogens like salmonella or listeria or contaminants like allergens or toxins. And maintaining rigorous quality control throughout the production chain, they give us confidence in our food supply.
That's a huge impact across so many sectors. I'm seeing more and more wearable tech in the news, like smartwatches and rings. Are biosensors truly moving in that everyday direction, beyond just medical clinics?
Absolutely. It's a major, undeniable trend. The future is integrated. Wearables and implants are truly where biosensor technology is headed for widespread adoption. The focus is increasingly on highly integrated, miniaturized, and even disposable biosensors for continuous, non-invasive monitoring. Lab-on-a-chip, LOC technology, is at the forefront of this.
Lab-on-a-chip.
Yeah, it integrates microfluidics, tiny channels to handle fluids, various detection methods, and nanomaterials onto a single chip the size of a credit card or even smaller. This enables multi-analyte, rapid, and cost-effective point-of-care diagnostics, bringing complex laboratory tests directly to the patient's bedside, the doctor's office, or even your home, enabling immediate results without sending samples away to a central lab.
Bringing the lab to the patient.
Exactly. We're seeing wearable biosensors becoming more common right now, such as smart wristbands with ion-selective electrodes that can analyze sweat electrolytes in real time, giving insights into hydration and fatigue during exercise, for instance. Even futuristic concepts like smart contact lenses are being explored for a non-invasive diabetes diagnosis by analyzing glucose in tear fluid. Imagine a continuous, discrete glucose monitor built right into your contacts.
That's incredible. A contact lens monitor.
It's being worked on. What's more, emerging manufacturing technologies like 3D printing are offering exciting advantages for biosensor production. Higher resolution for intricate designs, potentially lower production costs, and unprecedented end-user customization, making personalized medical devices a real possibility. However, despite this strong technology push and incredible potential, significant hurdles remain for widespread commercialization and everyday adoption.
What kind of hurdles?
Well, things like ensuring long-term stability in harsh biological environments, the body is a tough place for electronics. Effectively managing interference from other complex biological fluids in real time. Developing robust, reliable electronic signal processing for seamless wireless transmission of data. There's also the critical need for cost effectiveness, making these devices affordable for the masses, and generating genuine market pull, ensuring that there's a real demand and acceptance for these powerful technologies in our daily lives. The interdisciplinary nature of nanotechnology itself can be a challenge too, requiring diverse expertise across biology, chemistry, engineering, data science, all working together to bring these innovations from the lab to real-world, impactful products. It's an exciting but challenging road ahead. Outro.
So what does this all mean for you, the learner, who has now taken this, pretty deep dive into this complex but absolutely crucial field?
It means that biosensors are not just abstract scientific concepts confined to academic papers, right? They are tangible, powerful tools with profound implications for personalized medicine, for safeguarding our environment, and for enhancing industrial efficiency. The continuous drive towards miniaturization, the enhanced sensitivity that nanomaterials provide, and their increasing integration into everyday devices. It all points to a near future where real-time, on-demand biological information is not just possible, but readily accessible to everyone. Understanding these fundamentals and the diverse applications of biosensors provides a crucial foundation for appreciating how we're increasingly able to leverage nature's own ingenious mechanisms for detection and translate them into actionable data.
It's truly fascinating how we're increasingly able to listen in, aren't we, on the intricate biochemical conversations happening all around and inside us, translating those whispers into actionable data that can literally change lives.
Indeed. And this raises an important, maybe even provocative question for us all, for you listening. As these incredibly powerful diagnostic tools become more widespread and integrated into our lives, providing continuous granular biological data, how will this reshape our understanding of health, disease, and maybe even human potential? What new questions will we begin to ask about ourselves and our environment when we have such immediate and precise insights into our own biochemical states? The implications are truly profound.
A thought-provoking idea to ponder indeed. That's all the time we have for this deep dive into the fundamentals of biosensors. We hope you feel a little more well-informed, perhaps even more curious, about the tiny detectives working tirelessly behind the scenes. Until next time.